Hey everyone. Um, Before we get started today, I just wanted to say that today is Trans Visibility Day, and we stand in solidarity with our trans friends, family, and colleagues, especially as they're under attack in places like Arkansas. If you're not aware of what's going on right now, I'll drop a link in the chat. Please educate yourself. But today is a day also for celebration, not only for trans visibility, but also a lovely day for me, as I get to introduce Dr. Shaz Zamore. Today, I'm very excited to introduce Dr. Z. Shaz did their PhD work with Tom Daniels and Adrienne Fairhall at the University of Washington in Seattle, where they studied how mosquitoes use thermal cues to navigate plumes using a combination of computer vision, experimental data, and analytical models. Shaz then moved on to the Socha Lab for their postdoc, where they began their exciting work on flying snakes. There, Shaz continued to combine modeling, data analysis, and experiments, including my favorite, a closed-loop VR system for flying snakes. Shaz is now an instructor and STEM outreach coordinator at UC Boulder Atlas Institute, where they also continue to work on flying snakes, taking cues from diverse fields near and dear to my heart, including ethology and dynamical systems. And with that, I'll hand it over to you.
Awesome. Thank you so much for that intro. And thanks for, the support with trans visibility and trans support. It really is a daunting endeavor. But there is, like you said, a lot to celebrate. And I think one of those things worth celebrating is the people that work really hard to just make those changes. So I'm going to give this talk, at least started off in a somewhat unorthodox way. I'm going to just talk to you guys. I feel like a lot of folks tend to just kind of dive into the science, but I think who I am is really important to the story. It wouldn't make sense without it. And so I wanted to just give a little bit of background. So first, I'm so excited to give this talk. I was telling a friend of mine that this is the first time that I'm going to give a talk and actually touch on the discussion points that are in my recent paper, because I've never come to a place where people really think about ecology and the impacts on neural systems and neural processing. So just give you a little background. I did my undergrad at Cornell University. I majored in biology. They have a focus that you could take. So you're going to concentrate what you study. So it's like microbiology or evolutionary biology and evolution. And ecology. And I was really interested in two things. So all of the classes that I came to Cornell to take were in ecology and evolutionary biology, but then I stumbled across neuroscience. I thought that was cool. So I was like, I'm just going to take them both and have a double focus. So my undergrad and like really the basis of my scientific knowledge starts around looking at neuroscience with these ecological and environmental considerations. So everything that I studied, everything that I do is always framed in, well, what is the animal doing? What is the niche that it's occupying? What is it doing on a day-to-day basis? Because all of these things are going to reflect in evolutionary stresses, which will then influence how the nervous system will function. So and I'll try to smoothly switch to share screen here. But when I was studying mosquitoes and their abilities to fly and how they navigate using heat, I'm going to turn off my camera. Give me 1 second. Let me do a little back and forth for a second here. Right, so when we look at their flight trajectories, when I looked at the analysis, it turns out that these guys basically fly in a random walk. They have very little predictability. Sorry about the sound here. They have basically a very difficult, predictable path, right? And so when we first saw these random walks, we always said like they fly like they're drunk, like you don't really know where they're going. They have an upwind bias, but that's about it. And we were really scratching our heads as to what was going on. We were expecting them to fly a little bit more like Drosophila, where you see these very clear turns and turn events that happen, but they don't really do that. Their flight paths really do look a lot like the trajectory that you see on the screen there. But it turns out if you are trying to be a parasite and you don't want to get caught by predators, then you're going to want to have a very hard to predict flight. And so when we think about how mosquitoes live in their world, right? They're trying to get this food without getting caught, unlike Drosophila, which don't have this threat of being swatted by literally everything it eats. You start to get a little bit more insight into the behavior and then therefore into the nervous system. So when it comes to snakes, snakes are just, they occupy a very different place in my heart and mind. I am someone who very, very deeply loves snakes, and I can actually present this guy for you. So I've always been around snakes. I've always loved snakes. My parents didn't, so it wasn't until college that I started to get snakes. This is my ball python trouser. I think he's like 8 or 9. And then I have a boa who's much bigger than this picture now, even though I took that about 6 months ago. So I have lots of snakes, and then here's a flying snake sitting in my head. They really like to get up high and look around. So I've always...
Sorry, the slides aren't showing advanced.
Oh, no. Tragedy. Yeah, let's see. I might just turn off my camera. I'll try to pop back in so you're not just listening to a voice, but see what we can do. And then I'll share screen. All right. How about now?
Yep, that's
I need vocal content. Okay, cool. Awesome. So I love snakes. I've been around snakes for a whole long time. I like when they climb on me in particular. But what brought me really to snakes in a research fashion was thinking about their evolutionary history. So when we consider other, I like not just snakes, but scleroglossans. So that's skinks, monitor lizards. I think there's some glass lizards in there. But everything with a forked tongue, basically. When you compare snakes to pretty much the rest of reptilia, their eyes, they have a really weird evolutionary history, which reflects in their morphology, right? So let's look at a gecko, right? Primarily terrestrial, arboreal clade of organisms. And their eyes are a little bit different from mammalian eyes. They're a little bit closer to fish or birds, where they have this ectodermal conus, which is a main source of vascularization to provide blood and oxygen to the retina, which you see here on the back. They do have a fovea, and they have a lens. And much like humans...
We actually don't see that. Now it's just snakes. Yep.
Can you see it?
That works. Yes.
Oh, okay. So I think the problem is how I'm sharing the screen, not what is on the screen. I'm going to try to come back on. Forgive me. Bear with me. I think I have the solution here, which is just to share the whole dang screen. So now let's see. How about that?
See gecko.
Great. Excellent. Awesome. Okay, so geckos' eyes are a little bit closer to fish or birds, where they have this ectodermal conus, right? So this is a vascularization. We have a vascularization all the way across kind of regularly. They have slightly different, more metabolic demands. And so this physical structure has to be a little bit different to provide oxygen to the retinal tissue. So like other amniotes, so amphibians, mammals, etc., geckos have solved the problem of adjusting the focus of the image on the retina by deforming the lens, right? So we will squish the lens if we want to have a longer field focal length, and we'll expand the lens and squash it laterally if we want to have a shorter focal length. But snakes spent about 75 million years underground, and in that time, they basically evolved away their eyes. They had a really basic lens, they had a really basic retina, and that was about it. Spectacle over their eyes to protect it from all the dirt, but that was it. And then they became terrestrial again, and so they had to solve all of the same problems of how to adjust focus, how to provide oxygen to the tissue, as all of the other amniotes, but they had to do it again, and they had to do it under different ecological pressures, right? They're now in a very different evolutionary and ecological context. And so instead of deforming the lens in order to adjust the focus. For example, these guys will move the lens, just move it forward and backward to adjust that focal length. So we're already seeing, if we're looking at the primary sensors, a primary sensing organ, we're already seeing drastic changes between snakes and the rest of reptilia, let alone amniota. So as a scientist, I'm just like, all right, everything has to be different there. And what's fascinating about snakes is that they didn't just come above land. They came above land, like they brought it. So some of these snakes stayed underground, like the blind Texas snake that we see here. It was largely fossorial. You can see they don't have too much elaboration of the eye. And then we have a couple of species that have varying levels of aquatic living. So here's a water snake that's Neurodia and a sea crate. Sea crates are so adapted to being underwater that they cannot move on land. They've completely lost their ventral scales. They can't undulate. So drastic body formations to accommodate these different ecological niches. And then when we think about terrestrial snakes, we have lots of different substrates to move on, right? So we have the sidewinder that's known for moving on sand quite well. It's not the only snake to move on sand. The Kenyan sand boa, which is demi-fossorial, spends some of its time underground, or at least partially buried. And then the Galapagos racer, which is adept at climbing over rocks. If you guys have seen the new Planet Earth and watched the iguanas running away from the snakes, then you've definitely seen how well these guys are able to move over very diverse substrates. So rocks, loose pebbles, and things like that. And then they moved, of course, into arboreal spaces as well. Here we have the brown tree snake, Boiga, a Cuban cave boa. These guys will hang from the ceilings of caves and grab bats out of the air at night and at dusk. And then we have vine snakes, Ahechula, genus Ahechula, which are found in Indonesia. And this is actually a new species that was discovered in 2019. So we're still finding how diverse and expansive they are. So not only have they moved into keeping in fossorial, working with terrestrial, going into water, and then also coming above onto into trees, they've also, with flying snakes, moved into aerial space. I do want to say that the vine snakes, that last snake that I showed, is also aerial. They do a lot of jumping and lunging. They spend a lot of their time in the air, but they don't glide like these guys do. So here we're looking at Chrysophilia paradisi. This is the paradise tree snake. They're found in Southeast Asia, in the Malay archipelago, pretty much everywhere from the southern tip of India all the way out to the Philippines. And you'll notice that this particular snake is spending a lot of time taking a visual survey of where it's going to go before it goes into a J-loop, a jump, a dive, and then a glide. And I know watching it once is never enough, so we'll watch this again.
We lost your screen.
Oh, tragic. Can you see that? Do you see my screen at all? Weird. I don't know why that stopped. Aha. Very curious. All I did was go back on the video, and it apparently didn't like that. All right, let's try this again. Worth seeing. Okay, so hopefully, can you guys see that? Yes, hopefully.
It's like halfway down on the screen.
Okay,
sorry, I was muted. Oh.
Is that any better?
Yes.
Okay, so we can watch a snake here. It's taking a lot of visual assessment. It's going to go into a J-loop, launch, dive, and then glide off into the woods on the right. So hopefully you guys see. If not, I will leave you with there it goes, dives, and bye, see you later. So if you guys can't see it, I'll do my best to give you guys just a wealth of links so you can see these guys gliding. But what's interesting about these guys is that they have had extreme morphological adaptations to allow them to glide. So they're able to achieve gliding flight by articulating their ribs. They move their ribs from a kind of D shape where everything is nice and flat and linear, and then they expand it out and then slide it forward, as you see here on the right image. And this type of articulation is similar to what you see in cobras as they flare their hood. And again, if you look at the relaxed state, the ribs are back and down, and then in the expanded state, they're up and forward. And instead of doing this on just the top of their neck, the upper third or so of their body like cobras do, these guys evolved to do it from their entire length of their spine up until their tail. So they flatten from just behind their head, the very beginning of their neck, all the way down to their vent where the ribs stop. And as if you look at the cross section that they make as they're flattened out, it creates this airfoil, which produces a really nice lift to drag ratio, which is important for getting a long distance, glide distance, for how far that you'll fall. So let's talk about how these animals compare to other species. We're going to talk about the glide ratio. So every single organism that glides will move out a certain distance for a given height, right? And so we can look at what this angle is relative to horizontal, take the tangent, and we'll basically get the height over distance or lift over drag. And we'll use this glide ratio as a means of determining how efficient a glider can be, right? And so efficiency should always be couched in what the goal, the behavioral goal is, what the function of the behavior is, but we can look at it numerically and just make a comparison across species. So the higher the glide ratio, the farther you can go without losing that much height, right? So ideally, one can surmise that if you want to glide long distances, you want to have a very high glide ratio. So let's look at a couple of different species of gliders. First, one of the most adept gliders that we know of is the colugo. This is a primate species that it's quite large and has very, very exaggerated patagium. Pretty much anywhere that it could stick a skin fold, it stuck a skin fold. And these guys have a glide ratio of 12. They're on par with vultures, so very efficient gliders. They can go 12 meters out before they lose, only losing 1 meter of height, right? So very, very far. Then there's the flying lizard, genus Draco, and these guys do all right. So we'll see a drastic drop. Not everybody glides as well as the colugo. They're standout. They're very good. So the Draco is about 1.6. So it can go about 1.6 meters for every meter of height loss. Then we have the flying squirrel, Terramini, which is also found in Southeast Asia. And these guys are pretty good as well. There are a number of other gliding organisms. So there's Rhacophorus, the gliding tree frog, and they don't do very well. They have a less than 1 glide ratio. And then there's Tychozoan, which is a gliding gecko, and they also don't do very well. So these two are actually considered parachuters because they have these very low glide ratios. If there's math buffs out there and you guys are remembering, the tangent of 45 degrees is 1, right? So everything that's less than 1 is going to have an angle that's more than 45 degrees. So they're basically doing a drop instead of extending out. And so we don't really see, although I put asterisks here because they literally took these animals and dropped them from the sky. They didn't allow them to induce their own gliding behavior. So you're not really getting what their natural posture is and what they would do. So it's not very clear data. But overall, what we know of, what we've seen, not as good at going out as the rest of these guys. So where does the flying snake fall in all of this spectrum? So we looked at, this is with the Soha Labs work. This is a paper that came out in 2011. And they had dropped and in this case, we allow our snakes to glide. So we coax them to glide by tapping on a branch, doing a lot of predatory behaviors, doing a lot of kind of lunging and thrusting to encourage them to move out to an end of a branch and jump and glide. So we're trying to get them to induce their own postures and behaviors to glide. And when we do so, we see that they have approximately a 1-to-1 glide ratio. And there are some that do far better. The smaller the individual snake, the better glider, meaning they have a better glide ratio, higher glide ratio. So these guys, the flying snakes, end up in the same region of performance as a flying squirrel. And take a look at these two body forms. Very, very, very different body forms, both going toward this ecological behavior of getting around its environment by gliding. So this automatically makes me go, what is going on in this environment? So all of these species that I've shown here are found in the same forest, the same ecological area. So here are all of the gliding animals. So here's a flying snake, the colugo, the flying squirrel, the flying frog, the flying lizard, colugo, and then a bunch of different flying geckos. They're all found in the same region in Southeast Asia, that Malay archipelago, called a dipterocarp lowland forest. And this forest is characterized by dipterocarp trees, which you can see one right here. They're very, very tall. On average, they're about 40 meters tall, or about 120 feet. And they're about 10 to 20 meters taller than the rest of the canopy. So having these predominantly dipterocarp forest means that you end up with this very sparse canopy. So just looking at this picture, you can see there's a little road here, but you can see there's a gap in this canopy. There's a gap over here. Pretty much anytime you see these lighter, taller trees, those are those dipterocarps, you can see that they are creating this patchy, sparse canopy. And scientists believe that this is how all of these gliding animals came to evolve, that it just became much more energetically, or at least temporally efficient, to move around the environment by gliding instead of climbing down, risking getting eaten, and climbing back up and expending an exhaustive amount of energy. So all of these animals, despite their differing body types, all had to solve the same problem of how do I get way over there if there's no bridge to help me get there. And so when Jake and I were kind of setting up this problem and setting up this project, we were really thinking about what this ecological task is. What does it mean to glide? So we know in flyers and powered flyers, particularly in birds, that their ecology will change features of their visual system, how fast they can see, how broadly they can see around their head, how much of a binocular region they have, etc. And so a lot of those are dictated by how fast they move, what they're trying to look for, if they're trying to identify a moving object or a very specific shape. All of these things will have impacts on their visual system. And gliders are pretty quick, it turns out. So if we, again, look at all the gliders, not looking at our parachuters this time, but just the true gliders. So the colugo moves at about 10 meters per second when they're gliding. The Draco, that flying lizard, moves at about 6.8 meters per second. Couldn't find anything on the flying squirrel, but the flying snake actually, sorry about that. Let's move this forward. The flying snake actually is moving on with the same speeds as these other individuals. So they move at about 11 meters per second. When you compare this to powered gliders, there's a very broad range where it goes from 8 to 23 meters per second. And we see a kind of breakdown in the evolutionary structure. So a lot of songbirds, kind of like your smaller birds, will tend to be slower, including birds of prey, which is kind of weird. And if you come to the faster ones, those are things like ducks and the very big migratory movers, right? So these gliders are moving, have the visual demand that's akin to what a lot of birds species are experiencing. Sorry. I have this thing on my computer where if I touch the wrong part of my trackpad, it just refreshes the whole page, so my bad. Okay, so similar to powered flyers, and snakes, again, coming back to this comparative biology, these snakes are the fastest recorded moving snakes, the flying snakes. So on land, mambas are the fastest. They move at about 10 meters per second. I think the fastest that was recorded, documented, published was 10 meters per second, but we record these guys on average moving at 11 meters per second. So they're moving faster than any snake that we know of. So this automatically makes me think we have a whole bunch of different morphological adaptations for its environment. We are seeing a very unique demand on vision, and it hasn't been looked at, by the way. So nobody's looked at gliders and what the ecological and neurological demands on gliding vision is. So this is the first time we're looking at it, and we're looking at it in this animal with this super funky body form. It's elongate and it doesn't have limbs, right? So lots of good stuff to dig into here. So their natural history is unknown. We don't really know that much about them. They're very hard to observe in the wild. They live all throughout the canopy, and they're very, 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 very shy. They do not like to be seen. They do not like to come out. They're very difficult to observe in the wild. They're also very tiny, so it makes it hard to see. So I'm going to talk about some research that I did to establish some visual characteristics, particularly their field of view, their acuity, so their spatial and temporal visual resolution. You can think of this as the resolution of your screen, so how many pixels you have. The more pixels you have, the better resolution, the finer your image quality. And you can think of it like your refresh rate. So the faster you refresh, the cleaner of view that you get and the finer detail of motion you can detect. So if anybody is an avid gamer and you're out there getting those 120 hertz monitors, then you definitely know that that little difference, even though we cannot perceive 120 hertz, it will clean up what we can see so we have very clear vision. So the same thing happens in a nervous system, and I'm going to talk about what we saw in those two spaces. And then I'll finish by talking about some visual behaviors that I saw. These guys are very quirky. I think you're going to love seeing how they act. They're kind of like freaky little birds. So we'll talk about some visual behaviors and what implications those have. And then I'd like to close by doing a little bit of a participatory activity, if we can, where we will collectively make guesses about their ecology. So this is something Jake and I argue about constantly. What they do, how often they glide, why they glide, who in the species, male, female, young, younger, older, are doing most of the movement, what eats them, sorts of things like that. So I'm going to show you a bunch of information about their visual system, and then we'll see what we can infer about their ecology based on what we know about how they see. So let's start by looking at their field of view. So I'm a nerd. I love programming. I consider myself a neuroengineer. I really like the design of tools and using new tools to help explore neural space. And so to look at their field of view, I didn't want to do an ERG where I would have to sacrifice an eye or sacrifice an animal. And these are extremely hard to get. We have to go there ourselves most of the time and catch them ourselves and fill out a lot of paperwork and a lot of flying with coolers and angry snakes. So we don't really want to sacrifice our animals. So to figure out how they see, I did a digital version of something called inverse perimetry. So I took a laser scanner. From the laser scans of their head that you see here, that's my undergrad, Nicole, who was working with me, we created a CAD model, a 3D digital model of their head. And then using Unity 3D, I put the head in a giant sphere and I projected rays at it to see what position on the sphere can you see one eye? And then I did it for both eyes. So look at it one eye, stop, refresh, look at the other eye. So we get how they see, how they're able to see on this whole sphere. So to give a little bit more clarity, I basically had a little globe here that was shooting lasers, and it would target either one eye or the other and do a little sweep because natural light is not collimated, it's very scattered. And so in order to get an idea of what the snakes would see, we have to replicate the kind of light that would come to the eye. And so we just did a 45-degree angle sweep, right? Not very exhaustive, but just enough to give us a good idea. And then basically everywhere that this circle moved on every point of this globe, this pixelated globe, I looked to see which eye could be seen or if no eyes could be seen, meaning a blind spot. And so here's what we end up with. So on the left-hand side, we're looking at a globe view of what they're able to see. Everything in red is their binocular region where you see overlap of the two eyes' field of view. And then everything that's black is a blind spot, places where they can't see. This blue triangle indicates where the snout is, just to give you a sense of orientation. And right off the bat, we notice that there's a really big difference between male and female snakes. And I failed to get a picture, and I apologize, but the dimorphism between these guys is drastic, as it is with most snakes. The females are much, much bigger, which means that their snout gets in the way of their field of view of their eyes. So they have a much smaller binocular region, although they have approximately the same horizontal sweep around their head. We also see stark dimorphism in the vertical plane, where the females have a slightly up-tilted view and the males have a little bit more broader. And again, this is because the males have a smaller snout and it does less obstruction of the field of view, at least immediately in front of their face. So overall, we're seeing a fairly reasonable binocular region, far more than we expected for snakes, if I'm honest with you. And we see a very big, an individual eye has about, on average, between the two sexes, has about 130 degrees of field of view. So a very broad sweeping field of view, kind of like what you would expect to see in a prey organism. But we have this binocular region, which is indicative of a predatory organism, right? So we're seeing some hybridization between predator and prey. And if we look at this compared to other organisms, right? So let's face everything in the same direction. Let's compare it to humans. Let's compare it to owls. Let's compare it to pigeons. So we are primarily predators, so we have a very forward-looking field of view and very high binocularity. We see a similar trend with owls, which are very fierce predators, not a lot of predation. And then if we look at a more prey-like occupation, lower in the food chain, so to speak, we have the pigeon. And so we're looking, if we look at this comparison, we're seeing kind of something between predator and prey, right? So this is kind of falling in line with what we expect to see, or what the kind of place they occupy in the ecology, what kind of niche they occupy, right? So they're this weird hybrid, someplace in between predator and prey. And so from this data, we know that we have some idea that sexual dimorphism will affect vision. And so we suspect that males and females have a very different life history. So on a given day, male flying snakes are doing something different than female flying snakes. And what we know of in captivity is that the males in general are pretty active. They like to move around. They're much more wriggly when you hold them. They're much more bitey. The females are much more sedentary comparatively. Overall, they're very active snakes, though. I'll say they're like king snakes, very, very wriggly. So that's what we can tell from their visual field of view. But how do they process that? Let's look a little bit deeper into how their computing components of their brain work, not just the sensors themselves. So I characterize their vision using something called optokinetics. This is a very canonical way of looking at vision. It's very hard for me to find the methods to do this, because the last time optokinetic drums were really predominantly used were like the 70s and 80s, when we were like, oh, what do these animals see? What does a mouse see? We don't know. And now all of that research has really been exhaustively done. And very few people are looking at very novel systems. So there's not a lot of optokinetic drums. So I had to build this from scratch, figure out the design, test it out, and get animals in it. So here's this optomotor drum. On the outside, sitting on a turntable, are black and white vertical stripes. And I used black and white instead of sinusoid because of the way that ganglion cells are set up. And yes, snakes have ganglion cells as well. So just like us, they're very keen to edge detection. They're very keen to contrast. And so I wanted to focus on that instead of, I get into arguments about with computational neuroscientists about this all the time. But canonically, using vertical stripes instead of sinusoid is just fine for assessing characteristics. So around the outside, we rotate this visual scene, and the animal in the middle stays the same. And when we rotate it, we'll get a visual reflex called optokinetic nystagmus. And I'll show you what that is, and we'll talk about what that is in a second. But in order to elicit it, we can go from something very easy, so we have a very sparse spatial resolution with very broad stripes, to something more difficult where the stripes are finer and it's harder to differentiate the pattern. And we can change the rate at which this is rotated, which will affect their temporal resolution as well as their viewing distance, right? So if they're looking at something up close and it's moving really fast, it's going to have a lower temporal resolution than if you're far away and you're looking at something moving fast, right? So they're freely moving. So we had to add in a whole tracking component and factor in the distance at which they're viewing all of these features. But on the plus side, that means we can look at a broader range of distances and then therefore a broader range of spatial and temporal resolutions than if we had them fixed in place. You can imagine head fixing an organism without limbs is tough. We talked about it. We tried. We came up with some ideas, but we just let them freely move. So put them in this drum, rotate it at different speeds, change the different stimulus that they look at, and then look at how they react to it. So just a bit of forewarning, if you are visually sensitive, this will be dizzying. So if you close your eyes, I'll let you know. If it gets upsetting, I'll let you know when the video is finished playing. So here we have a flying snake in an optomotor drum. It's moving from left to right, as you see here. And you're going to watch the head track. And then if you follow the very tip of the nose, you'll notice a little tick behavior. And I'll call it out when it happens. There. It's very tiny. There. 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 And you do one more. Come on. Nope, maybe not. Okay, so we'll rewind and look at that again. So tick, tick, and stop. Okay, so that's it for the dizzying video. But if you didn't catch it, that's okay. We'll look at it in a different way. So we have these markers on the head, and we can track the position of their head relative to vertical on the video, just an arbitrary point of reference. And so from this, we can get angular position and then angular velocity. So if we look at their head angle over time, we can see in gray, these are that very slow phase of nystagmus, right? So it has two parts. So there's a slow phase, and then those ticks, that little hard to see nudge, are these little fast moments here. So not a lot of displacement and very, very fast. But if we look at the slow phase, we can use that for analysis. So ideally, if you're trying to use optokinetic nystagmus, you're trying to keep the visual scene fixed on the retina. So if you have a moving scene, you want to match the speed of that moving scene in order to keep that data or information constant on your retina. So we'll use this to make an estimate for how well they're able to track their visual motion. So we'll look at something called gain, where we look at the rate of the head moving and compare it to the rate of the drum moving. If it's the same, they're matched, then we say this has a gain of 1. And this is ideal, right? This is what we want to see. We don't want to be faster than how the drum is moving. We don't want to be slower than how the drum is moving. We want to try to match it as much as possible. If it's less than 1, right, then we have a gain of less than 1. This means that the head is moving slower than what the visual speed is. And if it's greater than 1, the head is moving faster than what the visual scene is. So I looked at this across 6 snakes and a whole bunch of different stimuli to see if we can figure out some tuning curves for where we see the strongest amount of response, the highest gain, and at what visual features we see. So what temporal frequency and what spatial frequency. And on the whole, we see that these guys have high visual acuity in terms of speed, but very low resolution. So let's talk about how we look at this. So if we look in a given degree of view, we would expect to see a certain number of repeating patterns, right? So ideally, we want to see 1 cycle in 1 degree. That would be 1 cycle per degree, right? And if we see more, so what we see here is 2 cycles. And let's say this is 1 degree. That's 2 cycles per degree. To give you a sense of scale, if you take your thumb and you stick it out at arm's length, that's about 3 degrees of your field of view, right? So 3 cycles per degree would be 3 cycles that fit in the space of your thumb at arm's length. So we looked at this, so we're looking at this gain, so how well they're able to match the rotation of the drum, compared to spatial frequency, which is calculated based off of their distance and the size of the stimulus, the size of the pattern. And we want to see, again, how many patterns fall into 1 degree of vision. And we looked at 3 different types of stimuli that go from easy to hard. And then the spatial frequency also goes from easy to hard. So the higher the number of spatial frequency, so like 10 cycles per degree, is harder to see. It's more challenging. It's denser than 1 cycle per degree, right? So if we look at this across the board, we see this very broad, flat tuning. We don't see very nice curves as we had expected to see. I'll give you an example of comparison. And don't worry if this isn't quite landing yet. I'm going to get there. I'm going to bring you home. Don't worry. So it's hard to kind of look at these and get a sense of where these guys see a peak performance. So we do see some arcs that are happening. There's kind of one that's happening in the dark blue. Here's one that's happening in at 36 degrees per second. Here's one that's happening there. Here's a very broad tune, right? But we're not really seeing something that's very crisp and precise. But if we look at the maxima, where the best performance is, the highest gain that we see across all of these stimuli, on average, we see that at about 2.89 cycles per degree. So we'll say 3 cycles per degree. That's terrible. Humans are about, oh gosh, I don't remember. I think we're about 10 cycles per degree. This is the equivalent of having 2,300 field of view. So in order to see, they have to be 20 feet away to see something that we could see 300 feet away. Not good. If they were humans, they would be legally blind. Then we do the same thing with temporal frequency. So we're looking at hertz. So how fast will a single cycle move across 1 degree of the field of vision? And again, we see these very, very, very broad tunes. And if we, the broad curves, sorry. And if we, again, look at where we see the best performance, where is the highest gain across all of these different experiments, we see that the maxima is about 46 hertz. We see up to 50 hertz, but we tend to, on average, humans are about 30 hertz. So they don't see with as high spatial resolution as we do, so they have a blurrier vision, but they see much faster than we do on average. I saw behavioral roll-off at about 80 to 90 hertz, so their upper thresholds are close to 100 hertz. So this is what we see with zebrafish, right? So these very nice curves that show these really clean drop-offs, and these do not look anything like it. And so when we make this comparison between other visual scenes, what we surmise is that these guys have very broad spatial and temporal tuning. So they're not trying to really hone in on having a system that looks at one range of spatial acuity or one tight range of temporal acuity. We have a system that's good at looking at all of that. Okay, so that was a clue. We stared at this for about 18 months. This was the bane of my existence because I couldn't figure it out, and I redid all these trials, and I tried to massage the data, and I'm like, this is what it is. This is what it looks like. What's going on? And this only makes sense when you look at their behavioral ecology. So they have this broad spatial tuning, they have this broad temporal tuning, but why? What is the benefit? Here's another clue. These guys have curiously low light level detection. If we put them in the dark and we slowly turn up the visual scene as that drum is rotating, so we can start to look at where we start to see first behavioral visual responses to motion. And across the board, whether we're looking at any response, like a start response or maybe tongue flicking versus nystagmus, where you see that slow and fast phase, or if we look at it together, there's no real difference. But across the board, their mean is 11.8 lux, which is about twilight. Sorry, I thought there was going to be another thing that came up. So they start to see this behavioral response at very, very, very low light, at about twilight. All right, so they have broad visual tuning and low light sensitivity. What the heck? Very strange. So just to recap, they have about a spatial frequency of about 3 cycles per degree. Again, if they were humans, they would be legally blind. Can't really do much with that resolution. And then they have a mean maximum temporal frequency of about 50 hertz, we'll call it, just for the sake of clarity. And then across the board, we saw generally low gain. We didn't see a lot of response. And this was also confirmed, the spatial frequency and the low gain was confirmed by Bruce Jane, who's at Case Western, and he also was doing the same kind of optokinetic work with a different species of Chrysopelea, which I also looked at but didn't publish on. And so we saw confirmation across 2 different labs. So they have low spatial acuity, that is definitely for sure. So who are these guys? They're diurnal. They have minimal iris constriction. They don't really change how much light comes into their eye, and they live in cavities. So they're going from dark areas to bright areas in the canopy without this ability to change the amount of light that comes into their eye. So having broad tuning allows for a very adaptive visual system, right? It's indicative of a very adaptive visual system. And it turns out that their visual behaviors are also indicative of that as well. I'm going to try to get through this quickly so we can have a chat about this. Sorry about that. This went the wrong way. All right. So these guys do a bunch of crazy visual behaviors that make them adorable, and I love them so much. So first, they'll track planes and birds that go overhead. They also engage in a behavior called a head wag, which you see here. It's kind of cut off, but this lateral oscillation of the head. And they'll do it while they're hiding in trees. They'll do it before they're about to cross a gap. Here they did it in the optokinetic drum, right? So here's this back and forth, side to side little wobble of the head. And so we call that a head wag. This was another thing that kept us stumped for a really long time. And then we noticed that they do this in another context, not just in the optomotor drum, but when they're doing this gap crossing task. So they go from one green branch to the other green branch, and we can adjust the distance between them and look at how they move across. And so when we compare these two behaviors, we get a little bit of an idea of why they're doing this head wag, this lateral oscillation. So we had two hypotheses. They're trying to induce motion parallax, or they're trying to visually camouflage with a moving background. So when we look at these two different contexts, so purple is in the optomotor drum and blue is going across these gaps, and we look at the kinematics, so we look at how their heads are moving in these different contexts, we see that the duration is very different. So when they're crossing a gap, it's on average about 5 seconds. When they're doing it in the drum, it's on between about 12 seconds. So they do it for a very prolonged period of time. If they were trying to glean distance information, you would expect to see some sort of extinction of the behavior when you don't get that verification. We didn't see that. It happens every single time, these very long periods of time, no matter how many times you expose them to the same stimulus. Sorry. I'm going to be more careful about where I hit my trackpad. If we look at the amplitude, no significant difference. So how much they displace their head from side to side doesn't really matter. But if you look at their frequency, order of magnitude different. When they're gap crossing, they move their heads very quickly at about 2 hertz. When they're in the drum, it's much, much slower, 0.2 hertz on average. So we are seeing this very, very, very different, strongly significant difference between these two paradigms. And from this, we can say, okay, we think we're seeing something that's varying with context. In other small jumpers like locusts and praying mantises, we see the same kind of head wagging behavior. And this is to induce motion parallax to help them gauge distance, right? So here's a quick video of that. Right, so that's swaying behavior, trying to get a sense of what's going on. And then we see this in snakes too. So
looks like your screen froze.
They'll do this little head wobble, usually before they strike their prey. There we go. Beautiful oscillation there. Do that dance.
The screen is frozen again?
Oh, no.
There we go. There we go.
Well, for the sake of time, okay. It's hard to see, but here we have a little garter snake moving, head wag, move, head wag, move, head wag. And this was actually written up in a paper where they only head wag when there's grass or other things that will move in the environment. And then we have a knoll, a type of a knoll over here doing the same thing. Climbing, wobbling, wobbling. Chameleons also do a very similar behavior. So in these two species, the Thamnophilus and Polychrys, this is a type of behavioral camouflage. If you are in a moving background and you stay still, you stick out. If you are moving with that background, now you blend in. So what we found was that these guys are head wagging to accomplish both of these tasks. So when they're trying to cross a gap, they're head wagging to induce motion parallax. And when they're hanging out in a branch or when they're in the optomotor drum and they have this whole scene motion happening around them, they're moving with it, so they blend in. So we have, again, more dynamics in this very dynamic behavior, very dynamic adaptations in this animal. I'm going to skip this for the sake of time because I would rather chat with you guys. So let's recap what we've seen here. So if we look at their field of view, we know that they have sexual dimorphism. So the sexes might have a slightly different life history. What they do on a day-to-day basis will vary. When we look at their spatial and temporal acuity, they have this broad spatial tuning. They don't see very well in terms of spatial, but they have very high visual processing. And then when we consider their visual behaviors, they have these very dynamic behaviors. So what I didn't show was that they do a dynamic behavior with their tongue. So instead of flicking it to pull in air around their head, they'll stick it out to fill the blind spot just in front of their head as they're going across gaps. Right? So if they're in this risky behavior, they can fill in that information loss by having tactile information instead of visual information. So what can we infer from their ecology? Just checking the time here. So these guys are diurnal. They are competitive maters. So this is one case study that came out. The gray is the female and all of the colored snakes are males. And so they do a sort of mating ball, kind of like mating ball light, where they have competitive mating, where many males go to one female. They also are predators. They primarily eat lizards and frogs, but they've also been, there's some lore that they'll eat bats and birds. And then they're also eaten by monitor lizards. I'll send you guys this link. So this snake has eaten a baby monitor lizard, and you don't think that it possibly could. But then monitor lizards also eat snakes. So it's this really cool reciprocal predation. And then they live at all different heights of the canopy. They're up at the top, they're down below, they find them in houses, you find them on electrical wires. They're kind of all over the place. They're very active and dynamic in where they are in their environment. And they use this, again, more dynamic behaviors, lots of lunges, lots of jumps to cross gaps, not just gliding, but they have kind of a repertoire of different behaviors for navigating through their environment. So what can we guess? We know some things about sexual dimorphism. We know from just handling these guys and having a colony that the females are more sedentary than males. I think that the males glide to females. Again, arguments that I have with Jake Soha. We know a little bit about their locomotive ecology. We suspect that they glide to escape from predators, although again, a fierce point of debate in the lab. And jumps are more frequent than glides. They're much more hesitant to jump or do what we call an escape drop, where they're not really trying, they just want to get away from you. They're not really trying to glide. They just want to get to the ground safely where you aren't bothering them. And then they climb a lot, and they're adept climbers. They have modified keeled ventral scales that help them grip, and they can climb upside down. They're excellent climbers. When it comes to the food chain, they're heavily predated, predominantly by hawks. We haven't had, this is again, just going on talking to locals and what people see in these environmental areas. And they're avid hunters. They eat a lot. And so they're always quite active. I'm going to skip through this. I don't think it's quite relevant. And that's pretty much where I want to leave it and just open this up to a conversation about other speculations that have come to you hearing this information. And I'll also take this time to field any questions. And so I'll leave this kind of on the side here and happy to chat in the last few minutes here. I can stay longer if people can as well. Yeah, so thank you for... hearing all about my favorite animals.
Thanks so much. Yeah, I think we also have had some success with continuing a conversation on Twitter, if that's okay with you as well. Maybe you can drop your links there.
Absolutely. Yeah, I'll put my handle.
Yeah. So Sama asks, great talk. I agree it's important to center the behavioral ecology of the animal. Can you say a little bit more about how we should contextualize the role of vision with the animal's other sensory behaviors, like heat sensing and flame flickering?
Yes. My gosh, you're my best friend. Yeah, so I've worked in a number of different systems. I've worked in the rat vibrissa system, the whisker system. I've worked with mosquitoes, and I've worked with snakes. And you really can't get to any meaningful insight about what the sensory system is doing without considering the whole ecology, right? But when it comes to, we kind of, I think just because of how our brains work, we love to get into this, okay, what's more important? What's more salient? What's the primary sensory experience? And I think that's a little bit of a trap. Because you have things like gating. So what we see in mosquitoes is once they get a sense of, they get a whiff of carbon dioxide, for example, their visual contrast sensitivity goes through the roof. And so in very few animals will we come across one sensory system that works fully independently of another sensory system. If you know about the McGurk effect, you know that doesn't happen in us, right? If we see vocalizations happening and different mouth shapes will perceive a different sound, even though the video is constant, right? So it's not so much thinking about what's the most dominant, although you certainly can ask, you can get some idea from like how much, real estate, nervous tissue real estate they have. But I think it's important to think about how they interact with each other versus which is the more important one, right? Mm-hmm. It doesn't really work that way. When it comes to thermal vision, in like pit vipers especially, that's so cool. So they actually have a lot of integration of thermal vision so it goes into the optic tectum. They have layered structures in the optic tectum where one layer responds to input from the eyes, one layer responds to input from the pits, and then you have oppositional. So if one group of neurons will only fire if you get both, or one and not the other, like kind of like on and off ganglion cells, right? So we see, again, this is more of the same, where we see like this integration and all these different sensory modalities really just feed into a whole sensory experience, right? It's not really about what's the one. I hope that answered your question.
Yeah. So Monica asks or says, "Thank you for the great talk. I was wondering, since the females can be more prone to jump away from predators," I'm going to paraphrase a little bit. Is there any difference between pregnant females, and maybe are they more aggressive or more sedentary?
Man, y'all are coming with the questions. I love it. Okay, so the females are more sedentary. The males are the ones that glide more. And when we see better glide performance, meaning they go farther for how far they are, the males outperform the females. It's night and day. We usually, when we're doing biomechanical flights, we're not even flying the females. We don't even bother, because the males are just so much more likely to fly, and so much, like, they'll go so much farther. So we get better data. So, I mean, we do fly, but we're just kind of like with the males. So when it comes to gravid females, good question. We just, the last few months that I was in the lab, we were trying to breed the snakes. And so we had 2 gravid females. They do not do anything. They sit very still, which is very common in snakes. They basically nest. We have a whole bunch of different nest sites. They pick a nest box and they stay there. If there is any food that comes around, they are ravenous. We had one female grab a mouse out of the mouth of a male. We were trying to mate them and we fed them, and we're like, okay. So whenever we have them to try to mate them, we don't feed the males, which actually is good for them. You don't really want to overfeed males. You get high fat and then low sperm count. Blah, blah. So you want the females to be very well fed, because it's such a tax. So I think that they're even more sedentary than they are when they're gravid. They're mean. They get real mean. They get real territorial. They come after you. As soon as they see you walk into the room, they're like, "What are you doing?" And as soon as you open the door, they're like, "No, we're fighting." So.
That's awesome. I think since we're at the end here, I think we will strongly encourage everyone to engage on Twitter. This has been super fun. Thanks so much, and feel free to drop any links. We'll retweet whatever you want.
Yeah, my pleasure. Thank you all for coming, and I'll hop on Twitter right now. So if you have questions, I'll answer them right away. Thank you, guys.
Thanks, everybody. You have a great day.